Marie Gibbons is here to talk about engineering clean meat. She is a researcher at Harvard Medical School, working with Dr. George Church on kind of the latest developments in clean meat. And she is going to talk today about the science of clean meat, where it's coming, or where it's coming from, and where it's going. And without further ado from me, I'd like to introduce you all to Marie Gibbons.
Thank you. Hi, guys. I also would like to point out that it was around this time last year that I was at EAGx Boston when I actually met with Dr. Church, and that is why I'm here now at Harvard. So you never know who you're going to meet here, what connections you're going to make. But yes, I want to talk to you guys today a little bit about how to grow meat, and I'll talk a little bit about my specific research as well. So first, I want to give you guys a general idea of what clean meat is. So it's a process where instead of using cows to make meat, or animals in general, we can actually replace the animal with cell culture, with scientific techniques. And just to throw this out here, this is not actually an animal-based burger. This is the Impossible Burger, which if you guys haven't gotten a chance to try that would be on burger. I highly recommend it. It's always a good time. So just throwing that out there. But the cool thing is that we've actually been replacing animals with scientific technology for quite some time. 100 years ago, as Bruce mentioned, we were very focused on using horses for transportation, and we now use cars. And similar to that, we also used to rely on oil for a light source, and now we have light bulbs. So let's talk a little bit about the light bulb of the clean meat industry. So this is a collage here of all the reasons why I'm really excited about this and want you guys to be too. So this first row here, again, Bruce did a really good job about talking about this, but basically animal agriculture is one of the leading causes of deforestation, of methane emissions, of desertification, of water pollution. Don't even get me started on what's going on in our oceans right now. And this is something that we need to address if we want to continue to live on this planet. The next thing I want to talk about is poverty, is the fact that we are letting almost 1 billion people go to bed hungry every single day. And our population is only projected to increase. So the fact that we can't even feed people now, that tells us something that we need to probably come up with some alternatives where we can feed more people, because we're going to have more people in the future. It would be awesome if a very high percentage of our population wasn't going to bed hungry. So some of the next few things are the fact that we could potentially make clean meat healthier than traditional meat. When you think about things like cholesterol or plant or animal-based fats, I'm not necessarily sure we'd be able to completely remove cholesterol from the product, but it's certainly something where we can remove animal fats and replace them with plant-based fats, which should, in theory, be a little bit healthier. The other cool thing that I want to talk about are these happy little blue rod-shaped things. This is bacteria that you get from poop, and it's the reason why you're supposed to cook your meat before you eat it, because it is generally covered in things like E. coli and Salmonella, which, again, are bacteria that live in poop. So, luckily, clean meat is going to have little to no legal matter involved. One of the reasons why it's called clean meat. So again, poop-free. I think that should be a very big selling point in the future. So the next thing is obviously animal welfare. That's something that got me involved in this, among many other things, clearly. But the idea that we won't have to raise and breed and torture and slaughter a whole bunch of animals just because they taste good, I think that's a really positive trait with clean meat. And then in addition to that, there's a lot of scientific benefits that go into accelerating clean meat research. Right now, a lot of our vaccines are being produced in eggs. I think that's something that can be scaled up, being produced more efficiently if you're growing them in things like bioreactors, which is something that we're already working on. And if that is the route that we go in terms of making clean meat, then those two areas of research can kind of feed off each other. Additionally, organ synthesis, regenerative medicine. Right now, I feel like the clean meat field is learning a lot from regenerative medicine, but as more funding and more research goes into clean meat production, we can start helping as well. So hopefully regenerative medicine can learn from us a little bit. And in addition to that, we can, hopefully if we're able to grow meat, if we're able to grow organs, then we can move away from animal fermentation in the labs. And that's, I would say, would be really beneficial for a lot of different reasons, not just for the animal ethics, but the fact is we aren't mice. And it's very possible that there are a lot of cures for a lot of really awful diseases that are sitting on a shelf somewhere because they didn't pass the mouse test, but they could work perfectly well in humans. So the more that we learn about how to regenerate tissues outside of the body and apply that to human tissues, the more accurate we can be in drug discovery. And finally, if you guys want to eat meat in space, you're probably going to have to grow it. So I think that's a big plus. Now that I've told you a little bit about what it is and why it's important, let's talk a little bit about how we actually make it. And in order to do that, we need to kind of dive into what meat is currently made out of. So this is a slide here of some of the different components that I can think of personally. I'm sure that there are other things in there that I don't know about yet, but just in terms of what I can think of. And the cool thing is these are actual pictures of the actual cell types. They're just very, very small SEM photographs that have been colorized, but here you've got your turkey leg, and then over here you've got skin cells, bone cells, blood cells, and then surrounding the blood cells I would imagine would be some sort of endothelial cell that makes up your veins and your arteries. There might even be some little macrophages here, your fat cells, your connective tissue, your neurons, and when you're thinking about muscle, and then of course your muscle cells, which is what I work on a lot. So I'm going to tell you guys a little bit about that. And I'm also going to talk a little bit about, well, not a little bit, I'm going to get more in depth with the process, the proposed process. So this is a diagram that the Good Food Institute put together, which involves the process of making meat. So you start out with cell culture, put those cells into a proliferative bioreactor. You're able to use media that can be recycled. From there, once you have enough cells to work with, once you have enough cells to make a burger or a nugget or whatever it is that you're interested in eating, you can seed them onto a scaffold, and from there you're able to get a final product. So I want to jump into each of these a little bit more in detail. So the first thing that you need to do is pick a cell type. I'm aware of 4 different cell types right now that you can use to grow meat. Again, there are probably more out there. But the first one would be embryonic stem cells. I'm not the biggest fan of that because I don't like the idea of taking cells from an embryo. But if that ends up being something that's the most efficient way of doing it, then I think that the benefits would outweigh the costs. So you have your embryonic stem cells, and then you also have your mesenchymal stem cells, which are very similar to embryonic stem cells, but you can actually get those from a biopsy from an adult animal. You can get that from fat, you can get that from muscle, you can get it from bone marrow. And so I'm a little bit more of a fan of the mesenchymal stem cells, but one of the benefits of working with the stem cell is that these guys can, they're pluripotent, which means that they can turn into various cell types. So it's possible that we could take one biopsy and differentiate these cells into all of those different cell types that I was talking about just a moment ago, which I think is pretty cool. What I'm personally working on is satellite cells, which is a muscle stem cell. So one of the benefits of that is that they are very easily nudged in the direction of turning into muscle, if that's the cell that you want to produce. And right now, considering the fact that the majority of meat is made out of muscle, that is something that most of us are focused on. And the other cool thing is because they are muscle stem cells, it's easier for them to turn into muscle. You really don't have to do much, just get them kind of crowded and take away their growth factors, and they'll turn into muscle. And so they have that benefit over the embryonic and the mesenchymal stem cells, because those guys usually take a little bit more nudging to get in the right direction, and sometimes they'll differentiate the things that you don't necessarily want them to turn into. So another possibility is the idea of induced pluripotency, and that's where you take a cell that has already decided what type of cell it wants to be. It could be a skin scraping or a cheek swab or the tip of your hair or a feather follicle, and you can actually tell that cell to turn into a stem cell. And again, it has some of the disadvantages, at least right now, in terms of being able to maintain that stemness and not getting it to turn into something else. But I think that there's a lot of potential for using this sort of technology. I also want to note that at the moment, the efficiency is highest when we are inducing pluripotency using genetic modification, but it is possible to do this through media additives. So if it's something where you are concerned about using genetic modification, induced pluripotency is still a possibility. It's just right now the efficiency isn't as high, which I would say would arguably be a reason to explore that more. Get those numbers up. So, in terms of growing the cells, I'll talk to you guys a little bit about muscle anatomy, because that is what I'm working with. So here we've got our turkey leg, and just a little crash course. I'm going to go ahead and take the skin off, because it's healthier, right? So if you were to take a look at this, do a cross section of this, you have a muscle organ, and within that muscle organ, you have a bundle of tubes that make up a fascicle. And then within that fascicle is another bundle of tubes. You guys are seeing the trend here, right? And that right there is your muscle fiber, which again is made up of bundles of tubes. But your muscle fiber is a single cell that's got a nucleus, and then you can also see here these little guys called satellite cells. And the role of satellite cells is actually to help muscles grow. So now I'm going to talk to you a little bit about the physiology of muscle growth and how satellite cells play a role in that. So when we are discussing muscle growth, unless you are in embryonic development, when your muscle is growing, it is due to an injury. It's due to a tear within the muscle. And that could be due to puberty, it could be due to you going to the gym and working out, it could be due to you going and falling down the stairs. No matter the reason, in the end, it is caused by a small tear in your muscle. And what happens is when your muscle gets this tear, your satellite cells get activated. So this satellite cell, which is usually just kind of hibernating, hanging out, waiting to be called into duty, it will be activated, and then it will start to proliferate. One cell will turn into 2 cells, 2 cells will turn into 4 cells. And this is what we call proliferation, which is rapid doubling, rapid growth of the cells. And then after there are enough cells to fill in that gap or add on to that muscle, make that muscle bigger, they're going to undergo differentiation. It's called terminal differentiation, and they actually fuse together. And they can either build onto that myofiber or make a new myofiber. And so that action right there is able to fill in the gap. So using that information, we can actually take the biology, take the research that has gone into how to grow muscle in the body and take it outside of the body. So that's what I did. I ended up using a biopsy from a turkey. And what you do is you take it from primary culture. And again, this doesn't have to be a muscle cell. There are several cell types that you can use, but I worked with a muscle cell biopsy. You place it into culture that mimics what the cells will be experiencing inside the body, inside of that muscle fiber environment. So the next thing that I did was a little strange, but I actually starved the cells. I gave them media without any growth factors in it. And what it did was it killed a lot of them, but it also selected for a very robust cell line that was able to live without these growth factors, at least in part for a certain amount of time. And I think it also had some effects on the genetic expression of the cell, which is something that I'm currently studying. And then from there, I did go ahead and feed them. And throughout the rest of the process, I did continue to feed them. I didn't start. I'm not that much of a monster. So what I did is I would feed them every 48 hours until they covered about 80% of the bottom of the dish, which is what we like to call confluency. And I was very careful to make sure that they didn't get too confluent, they didn't get too crowded, because you'll notice that if they get too crowded, they're going to start to form these kind of linear formations here, and that means that they're starting to differentiate into myotubes, which we want eventually. We want to be able to make these muscle fibers, but what I'm really focused on is not necessarily that part of meat formation where we make the meat. I'm more focused on making a lot of cells that can one day be used to be turned into meat. Which I'll touch on in just a second. So I made sure that they never really got, well, obviously they did sometimes, otherwise how would I have seen this picture? But most of the time they didn't get that far. Once they got crowded enough, I would use digestive enzymes to take them off the dish, and I would either freeze them down to create a stock so I could do more experiments later on, or if somebody was interested in using some turkey cells, we could send them their way. So if anybody is interested in some turkey muscle, I can hook you up. And then from there, I could also go ahead and repeat the process. So now that you guys know how to grow the cells, let's talk a little bit about the power of exponential growth, which right now is theoretical, and you'll see why in just a second. But the idea of exponential growth is that you have one cell, and then if this cell is able to double every 24 hours, the next day, you're going to have 2 cells. The day after that, you're going to have 4 cells, and then the day after that, you're going to have 8 cells. So, okay, big deal. You have 8 cells after 4 days, wow. But what about after 3 months? this is how many cells you would theoretically have after 3 months. I don't even know what that number is. But I'm going to try and put it into something that we can maybe wrap our head around a little bit better. So this is a little nugget that I make just kind of in my spare time whenever I had extra cells that I didn't want to freeze down. I would, throw them into the nugget tube. So this is one of my little micro nuggets, and it's around 5 million cells, and it weighs around 0.1 grams. Now, when you compare that to a MIG nugget, they weigh about 17.5 grams, which, if the math works out, that's around 875 million cells, which is a lot of cells. To put that into, get a better idea of that, the biopsy that I took from this turkey muscle was about the size of half of a grain of rice, and it was around 12 million cells. So this is a lot of cells. But remember those 90 cell divisions, those three months of growing these cells in optimum conditions, assuming that cell growth is the limiting factor, where again, we have 6.19 times 10 to the 26 cells. That's going to equal out to around 12.4 quadrillion tons of muscle. And if you wanted to look into the estimated requirements for the annual meat consumption by the year 2030, which is 376 million tons, we have the theoretical potential to make 33,000 times the annual global meat consumption, or the meat demand, in a 3-month period. So obviously that is something that we haven't done yet, but I think there's a lot of potential in harvesting that power. So the next thing that we need to think about is, why are we not able to grow this right now? I mean, clearly we wouldn't be able to grow this amount of meat because we wouldn't have enough to feed the cells. Everything, the entire world would be made out of turkey. But one of the things is that we can't grow, we can't meet the annual global meat demand using petri dishes. So this image over here, this was just a fraction of the petri dishes that were used to make the burger that was cooked and eaten in 2013. But luckily, there are a lot of different options in terms of how to grow these cells. My money is in these stir tank bioreactors. They're basically giant kegs that have little turbines at the bottom to kind of keep the cells moving around, keep the nutrients distributed. But there are other options as well. A little bit more intuitive process would be a membrane reactor, where you would take the cells and have them attached to a type of membrane here. I think that's what's going on here as well. You also have this kind of pillow-looking thing where the cells are floating around and they just get like rocked, which that sounds fun. And then there's a company in Israel that's talking about like this all-in-one meat-making machine, like an easy-bake meat maker. I have no idea how they're doing it, but I'm super excited because, I mean, I love that idea. You have your dishwasher, you have your laundry machines, you have your refrigerator, and then you got your meat maker. Like, I'm ready for this. So, once you have your cell type, and once you decide what type of bioreactor you want to use to grow your cells, the next thing you have to think about is what you're going to feed your cells in order to get them to grow. And that's what I'm really focusing on, which is the media, which is cell food. So when you look at the components of media, you'll find that it's basically protein powder and Gatorade. That's really all it is. Unfortunately, at least in academia right now, a lot of the components are also reliant on animal serum. Fetal bovine serum is a very popular additive. I do want to note that there are a lot of serum-free media options that are designed for specific cell types, specific cell lines. I'm pretty sure that there are several clean meat companies that have already gone serum-free. The issue is that they can't really talk about how they're doing it. So one of the things that I'm working on right now is kind of this general serum-free formulation that I can tell everybody what's in it, and then everybody can use it, and we can all, eliminate animals from this process altogether. Because obviously we can be growing meat for using cow blood to do it. If we wanted to make this process animal-free, which obviously we do. So luckily, there are options in terms of the serum-free media additives and the formulations. Right now, we're reliant on recombinantly produced growth factors. And so what we do here is you take a gene. Here in this example, they're using insulin because that's actually how we get insulin now. We used to take it from pig pancreas, and people would inject that into themselves, but now we use recombinant protein technology. What we do is we take the gene that is responsible for making this protein, we put it into E. coli, which is a really fast-growing bacteria. When used appropriately, can be good. You don't want to be eating it if it's coming from animal poop, but again, you can be using it appropriately to make things like recombinant proteins. So this bacteria, which grows really, really fast and is really good at making proteins, will make this insulin for us, or whatever type of growth factor that we want to use. And then we harvest it downstream and give it to the cells, or give it to somebody who needs insulin. And then these guys will grow in bioreactors. I know what you're thinking. Okay, Marie, you get it. You like to start saying bioreactors. They're pretty cool. But I think that we can actually make this process even more efficient. I think the idea of taking a sequence and putting it into an organism that doesn't normally make it, and then harvesting that sequence later on just to give it to something, not the sequence, the protein, I think that's something that we can, there's some tweaking that can be done. So there's some options. There's some people that are exploring the potential for plant-based proteins, kind of putting these meat cells on a vegan diet. The other option is when you think about what's in bovine serum, when you think about what's in the serum, it's not like some magical fairy is coming and waving her wand and bam, these baby cows have these magical proteins in them. These cows are made up of cells that are in turn making these proteins. So we can actually take the cells, this is an image of a macrophage that's responsible for making some of the growth factors that are needed in order to grow muscle. We can take these cells that naturally produce these proteins and grow them as well, and then use the proteins from that to feed to our muscle. But I think something that's even more efficient than that, and therefore would be even cheaper in the long run, would be to actually genetically design the cells that we plan to eat to make their own growth factors, to feed themselves. And so that's where the genetic modification, potential genetic editing plays a role. So I want to talk to you guys a little bit about that. Here is a very basic overview of what's going on whenever a growth factor interacts with the cell. You have your ligand, which in our case would be the growth factor. And then you have our cell, which would be the muscle cell. And on this muscle cell is a receptor. So just a protein, just a sequence of amino acids put together in a very specific architecture, sitting in the membrane of the cell. When this growth factor and this receptor interact, bind, it causes a signaling cascade within this cell. And this cascade causes a response. And our situation, that response would be growth. So I want to talk a little bit about how we can learn from this process and what we can do to manipulate it. But first, I want to give you guys a very basic overview of something that we like to call the Central Dogma. And this is the idea that you all start out with DNA, the specific sequence within DNA, then gets turned into RNA, which in turn gets turned into protein. It's very basic, just in terms of a visual. Here's your cell, the nucleus. Inside the nucleus is a whole bunch of DNA that's packed up into little chromatin. And then from there, you've got your DNA, which gets translated or transcribed into RNA, which in turn gets translated into protein. And then this protein can be used within the cell, outside the cell. It can be a growth factor that is excreted outside of the cell and interacts with another cell. And here, I like this idea here of, maybe this protein is a growth factor receptor, maybe it's a growth factor itself. And so there's where it really plays into how we could actually make this process even cheaper. So we could have these cells making proteins. Maybe this cell, instead of waiting around for this growth factor to be added to the media or created by another cell, maybe this cell just makes this growth factor itself. Or even more so, when you look at the different proteins that are being changed within the cascade, what if we make it so that the cell produces the protein in the specific architecture, in the specific sequence that is needed in order to elicit a response? Or even further, what if we went to the genetic expression that the response is responsible for and just make it so that is always on? So as long as these cells have access to the building blocks needed in order to make more cells, they're going to make more cells without any need for signaling from a growth factor. In terms of the tools that I'm working with, I can't really I don't think I have time to get into this too much, but if you guys ever have any questions about it, I love to talk about it. But this little guy here is Cas9. It's a protein that's used in CRISPR technology, and we can make very specific edits within the cell. We can do so via knocking the genes out, so making it so that certain genes no longer are expressed. There's also a technology called Viper, where we can do the opposite. We can tell a certain gene to get turned on. It's kind of like this protein will bind to the DNA and then kind of wave at the RNA machinery and say, hey, come over here, transcribe this gene for us, hook us up. And then the other possibility is that we can actually make specific base pair edits. So instead of knocking out a protein or knocking out a gene or turning it on, we can change specific nucleotides within the DNA. Another thing that I think is worth pointing out is that these two, well, for sure using CRISPR as a knockout tool, and I would also argue for this base pair technology, because this is something that can happen naturally. It's something that the USDA has in the past decided not to regulate as a genetic modification. So there are, I believe, 30 different plants in the market right now that have used CRISPR technology but are not labeled GMO. So I think that's something to take into account in terms of regulations and consumer acceptance. The other option, if you don't care about GMOs, is we can go ahead and just put the gene right in there, which is definitely a genetic modification. But I think also in terms of the research, I'm a fan of because it helps me figure out what I need to do to change the cell down the line, if I wanted to use CRISPR or not. But at the same time, if people are cool with genetic modification, then this is a really easy way. We can just throw the growth factor sequence into the cell rather than into the bacteria. Another cool thing is that there's something called a transposon where you can insert a gene into the cell, and then if you don't want it in there anymore, you can just take it out, which I think is pretty cool. And then another thing I'm working on is the serum-free media formulations, are those specific growth factor ratios that are necessary for the cells to grow without any animal products. And the cool thing about that is, one, it helps me identify some of the genes that I need to upregulate within the cell via CRISPR or transfection. But at the same time, it is technically GMO-free, I believe, because while the proteins, the growth factors themselves are being produced through genetic modification of E. coli, of this bacteria, the proteins are not genetically modified, the end product. But here's one thing that you really need to consider. If you are anti-GMO, you have to understand that you are giving up the potential to eat glow-in-the-dark turkey nuggets. So just want you guys to understand the sacrifice that you're making. I'm just kidding. I'm not planning on starting a company on glow-in-the-dark meat yet. Who knows? You like a niche market, right? So with that, I want to get back to the process of growing meat. The next thing that you need after you have your cells and your bioreactors and your media is your scaffold. So here are just a lot of different options that we have to work with. I personally really enjoy the idea of being able to use raw plant materials and combine those with turkey cells or any sort of meat cell that you want to work with. Things like coconut, jackfruit, artichoke that already kind of have this meaty texture. I think that takes a lot of processing out of the entire process. But we also do have the option of using synthetically derived scaffolds. When you look into regenerative medicine, organoid synthesis, people have put a lot of work into being able to create these scaffolds that are able to work within the body. So we have options there. Another cool thing is that when the cells in suspension, when they grow in suspension at least, they're actually able to produce their own extracellular matrix. So I think that's really cool. We might be able to harvest that protein and use some sort of synthetic technology to create scaffolding from that if people want a 100% animal-based product. And then we also have the option of extrusion, either by doing something where we would combine a processed processed soy or processed wheat, combine something like that and send it through the extruder. Which for those that don't know what an extruder is, you guys remember when you play with Play-Doh and you have those little plastic things that have like a mold and it would be star-shaped or heart-shaped, and you put it through that and you turn the. I feel like I didn't do a very good job of explaining that, but you get what I'm saying. That's basically what we're working with is the Play-Doh machines that you play with. So the idea is that you take something and you send it through this extruder and get something that has a very meat-like texture, which is very similar to how we get things like ground meat today. It's just that we send it through a grinder. So you're taking something that's more more structured and making it less structured in this sense here, versus here we could combine the muscle cells, and we could actually, you see here, these are little micro nuggets that I'm growing in suspension, so it's possible that we could take these guys and combine it with a plant-based product, or even on their own, send it through the extruder and have a meat have a textured meat product. Which brings me to the conclusion, which is the final product. So I just wanted to show you guys some of the current prototypes that we have. We've got chicken, we've got pork, we've got duck, fish, beef, meatballs. I'm not sure what that is, but I know it's going to be good because it's coming from Just Foods. Oh, and then obviously turkey, which is my favorite, but again, I'm a little biased. So that is what we have right now. So what I have to say is this is coming. It's already here. It's just a matter of scaling up. I hope you guys are ready. It's coming, and I'm really excited. So thank you very much. You guys
Thank you, Marie. Now we have a few minutes, I'd say 6 or 7 minutes for questions. And just if you can come down to the question mic, I believe it's on. Whoever gets you first question.
And it'll take about 2 seconds. I think probably everyone was thinking it when it happened. The phosphorus in turkey, if you eat a lot of it, do you get phosphorescent?
Oh,
Do you
you
glow
mean
in the dark?
the glow in the dark? I mean, so far I'm normal colored. No, I don't. I do not know. It's actually green fluorescent protein from jellyfish. So if you know of anybody who eats a whole bunch of jellyfish, then maybe. Yeah, worth looking into.
This is good.
Yeah.
Wonderful work. Congratulations. Can you tell us a little bit more about when you think we can expect these and at what cost?
Yes. So I am not working for a company, so I don't really know the cost, and I'm not really sure of the timeline either. But I would personally be pretty disappointed if we didn't have something by the end of this year, and definitely within the next 3 years. But my money is on by the end of this year, we're going to be able to buy something that has been made without animals, that is made out of animals. In terms of the cost, I would imagine that it's probably going to be 2 to 3 times as expensive as what you would expect from something that you get from a factory farm, but that's just the initial cost. And as we scale up, not only do we have the potential to be cost competitive, but we have the potential to be cheaper. I'm fully confident in that. So.
Hi. What kind of involvement in the clean meat industry is possible for people without a bioengineering background?
Yeah, that's absolutely great. Great question. So if you don't have a bioengineering background and you're not interested in getting one, I would say that consumer acceptance, getting into the business field, the policy that surrounds this sort of thing, the psychology behind it, all of those things would be really, really applicable.
One of the evils of factory farming you described is that we rely on antibiotics to clear away all these food bacteria, and that can drive the emergence of resistance. But imagining a future where we have just all of our meat growing in vats, of course I'm imagining there are risks there as well, both from bioterrorism targeting these cells which don't have immune systems, and also needing to pump a lot of antibiotics in there to make sure it's a sterile culture. Have you guys thought about protecting the meat and making sure there's no contamination this way?
Yeah, so I haven't personally thought about the potential for bioterrorism, but in terms of protecting the cells not using antibiotics or using a smaller amount of antibiotics, I think in terms of the process of growing these cells in large reactors, it involves a lot of autoclaving, it involves a lot of really sterilized techniques that if we end up using antibiotics, it would be at such a small scale that I don't think it would be as big of a deal. And the other thing that I want to point out is it's not necessarily the use of antibiotics that's so dangerous in the farm animal situation. The issue is that we're using antibiotics in an environment that is just the perfect breeding ground for more antibiotics. So if we are removing the fecal matter, the dead and the dying animals, then there's going to be a lot less room for bacteria to grow and therefore evolve into these super antibiotic-resistant bacterial forms. So I would say, yeah, there might be some antibiotic use. I'm personally not working with antibiotics. I think if people are careful about this, and then we also do have a lot of large-scale cell production for therapeutics at the moment that we can look at for in terms of models.
So my question is, as far as I know, in terms of meat replacement, the one approach is what you described, growing meat in the lab. The other, I think you mentioned at the beginning, Impossible Foods, doing plant-based. Do you see this two going forward as being complementary or kind of competitive? in the long run? Will certain people prefer one, do you think, or is those?
Yeah, absolutely. So I definitely see these as not competitive. I see them as complementary. I think, it's possible that they can be done together. I think a lot of the first hybrids or the first products will be hybrids. And I think, I really believe that plant-based meat over the next few decades or so is going to be able to target the majority of our population. But I also feel like there is a small population of people that, for whatever reason, they have that mentality where they want to eat animal meat. And I think that's where clean meat comes in. I'd also like to think of clean meat as kind of a stepping stone for getting more people to be content with plant-based meat as well as we continually work on making plant-based meat even better. So 100% we're in this together.
Our last question?
Sure. You mentioned quickly jackfruit. Was that to be used as the scaffold, or I missed the?
Yeah, that's to be used as the scaffold. It's got a very meaty kind of pulled pork look to it. So.
On second thought, we can touch both on or if there's not, we can just all take a break.